So, hi everyone, and welcome again to yet another session of Alberta MS Network Summer Lecture Series. We have Dr. Deepak Kaushik today. Dr. Deepak Kaushik is a postdoctoral associate at the University of Calgary. He did his PhD from National Brain Research Centre, India, before joining the lab of Dr. Young at the University of Calgary. His current work focuses on understanding the metabolic requirement of brain-infiltrating immune cells in the context of MS. And today he will be talking about the lifestyle factors in MS. So thank you, Deepak, and looking forward to your talk.
Yeah, thank you so much for this opportunity. It was really good to hear nice words from Neetu. And I'm sure you will agree with me when I say that this lecture series has been an enriching experience for senior researchers as well as younger trainees alike. And it is a pleasure that I'm going to talk about lifestyle factors in multiple sclerosis in today's lecture. And I know this the concept of lifestyle factors and environmental factors did come up in multiple talks previously, especially in the context of epigenetic changes and clinical epidemiology of multiple sclerosis. But what I'm going to do today and talk about basically is present evidences in favor of how lifestyle factors are indeed crucial modifiers of multiple sclerosis. And in doing so, during the course of my presentation, I'll emphasize on how environment interacts with genetic components and how this shapes of MS pathogenesis, and also would bring key mechanisms associated with those lifestyle changes and how important role do they play in the course of MS pathogenesis. So what have we learned so far? Our take homes from this lecture series thus far, and this is precisely what we know generally in the MS world. There is genetic association for MS. There is critical linkage to gene components in multiple sclerosis, which we all know, in the form of HLA, in the form of single nucleotide polymorphisms that associate an individual towards increasing risk of MS. And a lecture by Dr. Stephanie King from Lethbridge also showcased how epigenetic or post-transcription changes to genes can render or bring about changes to immune component, and that when interacted with other components such as lifestyle factors or environment poses a bigger risk for the development of MS. So this is precisely what we understand of MS, and I thought I would just put it out right in front to emphasize on that there is a critical linkage association between genes as well as environment, and environment shapes of genetic component by bringing about possibly epigenetic changes. Also to remember from the previous lectures that there's a critical window wherein these changes brought about can have effects in the future course of disease pathology. What do we understand of genetic factors? It often keeps coming that the genetic factors such as HLA typing is important. I thought I would just brief out very crucial, very highly associated or highly looked into HLA types. This HLA DRB1501 is associated with increased risk of multiple sclerosis, and it is a class variant of MHC molecule present on antigen presenting cells, as can be seen here, which presents peptides to CD4 T cells. And this renders an individual susceptible to increased risk of MS. On the other hand, if an individual lacks HLA-A02, that's what brings about increased susceptibility, because the variants of this gene class present antigens to CD8 positive T cells, which are important for immune suppression. So presence of HLA-DRB1 and lack of HLA-A2O2 brings about risks for developing MS. Again, these are not the only HLA types. There are several other, and depending on the population, one or the others can be enriched. For example, Spanish population may have other HLA typing as opposed to these ones. Studies such as genome-wide association, studies by looked at non-HLA single nucleotide polymorphism. These were found in patients closely associated with genes that regulate adaptive and innate immunity. And these evidences bring forth the idea that yes, immune component is very critical and probably even a driver of the disease. Although I would probably resort to saying that immune-mediated disorder rather than MS, immune-driven disorder. What are the factors that, that convey the idea that genes are involved? And you must have seen some of these data sets earlier or graphs earlier portraying how lifetime risk for MS increases as you start sharing your genes or genetic composition with a person who has MS. Right, for general population, while the lifetime risk stands at 0, which is very minimal, it starts increasing as you start sharing or you start closely associating with an individual, and it's as high as about 30-35% if you're a sibling or if you're a twin and a sibling who has MS. So monozygotic twins have 100% genetic sharing, and they have about 35 to 30 to 35 lifetime risk of MS, which means that there's clearly a genetic component associated with lifetime risk of developing multiple sclerosis. But what about the rest 70 or 65% of the twins? Why do they not all succumb to this disease pathology? And that's where the notions and hypotheses around non-genetic factors or environmental factors come into picture. That's when we start to dig in more about how environment shapes up immune component, how other lifestyle factors. One of the strongest proponents in this line of evidence for non-genetic factors happens to be migration theory. It is proposed that migration from a low-risk country for MS, in this case, to a high-risk country puts an individual at higher risk of developing multiple sclerosis. And it is also noted that migration that has happened around adolescence or before adolescence increases the risk exponentially to these migrants, as opposed to when migrants to these high-risk countries move at a later stage of their life. This is one. The second being, there has been significant rise in MS incidence in the last 2-3 decades, which cannot solely be attributed to genetic factors. And that's when, again, the whole idea about modifiable factors come into picture. And as of now, I think age, gender, and genetics stand uncontrollable. That is something that we cannot have over control over. But importantly, there are other risk factors in addition to genetic composition, such as smoking, physical activity, vitamin D exposure, hygiene, diet, the amount you sleep, or other infections. So all these risk factors are important to consider for a disease pathology such as MS, because majority of these lifestyle factors positively impact immune activation. There are some which are negatively associated with immune activation, such as vitamin D. But overall, it is critical for disease that has a high degree of immune component to it, such as multiple sclerosis. That is the reason why we and the field is highly invested in these ideas. Important to note that these individual risk factors, when added together, impose even a bigger risk, which is known as interaction effect. So if one is exposed to risk factor A as well as B, the overall risk is not just A plus B, it would be way higher. And I would just describe this in the next couple of slides as an example while I'll be talking about smoking. So this is just to be kept in mind that multiple risk factors eventually come together to impose even a bigger risk factor to the development of multiple sclerosis. So as I'll move the next few slides, I'll go and in a sequential fashion, try to bring in individual risk factors. To begin with, I would just like to talk about smoking and multiple sclerosis. And why is this critical? Why is this lifestyle factor is associated with multiple sclerosis? Here is a hypothetical situation where smoking, which is known to irritate, which is a lung irritant, can irritate lungs, can lead to chronic infections, and by process of citrullation can modify peptides, which then can activate some of the innate immune cells or even adaptive immune cells. Now, I've talked about how having HLA-DRB1 can predispose a person towards developing MS. These people who constantly smoke are then at higher risk of developing MS. And it has been noted that there is an interaction effect between these two. For example, while having HLA-DRB1 and lacking HLA-O2, the odds ratio are only 5. In smokers, which generally have higher, but way higher, way lesser than the risk factor put together, when they are combined, the odds ratio are as high as 14, which is a huge jump in terms of risk factors, suggesting that if a person who is already susceptible or genetic composition makes him more susceptible, indulging in habits such as smoking can increase the risk of developing MS even far more, and then probably the prognosis is far worse. What are the evidences in favor of this? In this study that was published in 2015 by an Argentinian group, went on to show how smoking actually worsens MS prognosis. Now, they had studied 2 different cohorts of people, one with people who were clinically isolated syndrome and looked at their conversion to clinically definite MS over 3-year period duration. And the second one recruited patients which were RRMS diagnosed initially and looked at their progression over time. And they categorized these 2 into smokers versus non-smokers. What they found was, if you look at panel A, that smokers, about 80% of smokers, CIS patients, converted to clinically definite MS as opposed to only 40% of CIS patients who were non-smokers, suggesting that the prognosis of smoking can be bigger risk factor. At the same time, when they looked at people who progressed to secondary progressive MS in the RRMS who were smoking, they found that this cohort had far fewer patients who did not progress, meaning that there were far more people who progressed to the later stages of the disease when they were smoking. Again, just pointing out that smoking is related to MS prognosis and can not only lead to initial conversion from an isolated episode to multiple sclerosis, but also has repercussions in progression of the disease. Other studies have also pointed out that smokers of both sexes have increased risk of developing MS regardless of gender, and it increases with cumulative dose of smoking. What does smoking do actually is the real question. So smoking, while we know increases Th1, Th17 responses, it also goes on to decrease T regulatory population in MS patients. It was the same Argentinian study where they looked at Treg populations in blood of smoker MS and non-smoker MS patients. They found that Treg population was far fewer. And overall, just to give an idea that the setting was skewed towards more inflammatory, wherein there has been, they noted that certain pathways such as indolamine dioxygenase pathway, which is relevant for production or the modulating IL-6 production was decreased. Therefore, there was increased production of IL-6 in these individuals. At the same time, these individuals produced more IL-17 and IL-22, giving an idea that yes, indeed, smoking has effects on immune system. If one thinks that smoking directly is bad, and if passive smoking is all right or vaping is all right, think again, because passive smoking is also associated with increased risk of MS. And I think it points out to the whole core idea about smoking as a lung irritant, rather than even low doses can be probably toxic. So this Swedish study looked at about 695 or 700 cases of non-smokers who never smoked their life and asked a question if they were ever exposed to smoke at home or at work, and then categorized these people into different categories as if they were never exposed, or if they were exposed less than 10 years or greater than 20 years, for instance, and then looked at their odds ratio of developing multiple sclerosis. When adjusted for other confounding factors, they found that the odds ratio increased, suggesting that the probability of these patients increased when they were exposed to smoke. That is, even passive smoking is now highly correlated with developing multiple sclerosis in these patients. So as a statement, I would just like to say smoking is correlated with increased MS risk. The other risk factor that the field is heavily invested in and has learned a lot is vitamin D3. And it is important to consider that vitamin D3 is not only relevant for multiple sclerosis, but also for conditions such as cancer, for instance, and bone malformations. For example, Western world had high incidence of rickettsia when vitamin D supplements were introduced in 1930s and 20s. They went on to decrease the episodes of rickettsia. But even then, there has been never uh, uh, an association with vitamin D exposure to decrease risk of MS with the supplements that were already provided. But yet, observations such as this are portray that that vitamin D could be affected, could be associated with the prognosis of MS. Now, the reason why I have this slide here is just to emphasize that there is clear demarcation in the countries which have more MS, notice here. So the prevalence, for instance, is very high in certain European countries as well as in the American countries. And if you go down south, Australia stands out in having more cases, and also countries like Argentina. But if I draw an equator line here and take you to the little geographic lessons and try to map out the region which have less incidence or less exposure to sunlight, these are the regions that will easily map out. These are the regions which generally are exposed to reduced UVB rays, and probably because they're away from the equator. And this is important because UVB is a critical factor needed for conversion or production of vitamin D3 in individuals. It is hydroxylated from 7-dehydrocholesterol. It's produced within the skin, converted to hydroxy vitamin D, and then in kidney, further hydroxylated to produce the immunologically active form of vitamin D. This is what has been studied the most. And why vitamin D? Vitamin D, we know it is a very important factor in regulating innate immune responses. Vitamin D binding to its cognate calcitriol or vitamin D receptor or VDRs can bring about changes in the expression of cytokines. For example, if you look at dendritic cells, there is decreased expression of IL-12 in these cells and increased expression of IL-10. Similarly, in interferon, in memory T cells or B cells, overall there's propensity towards generation of anti-inflammatory phenotype in these cells. Overall, this suggests that vitamin D3 can modulate immune responses. And what are the evidences that if vitamin D3 would it confer protection in animal models? It does. What we have noticed that when vitamin D is injected in these animals, it was noted that these mice, these EAE animals, do not manifest disease severity until at least day 20, meaning that probably vitamin D, by working at it, by modulating immune system, can contain something as severe as EAE. Now, it is important to talk about immune mechanisms. What about its role in demyelination or remyelination? It was also noted that in another study, when high dose of cholecalciferol or vitamin D is given in a cuprizone model of remyelination, it was noted that the exons were far more preserved when animals were given high doses of vitamin D3 in their diet, suggesting that it works on two different pathways, wherein on one hand it probably interferes with immune activation, and on the other hand it probably promotes something very critical for MS, such as myelination. What are the evidences in human studies? Are there in human studies that propose the idea? And there have been several nurse health studies from Harvard cohorts, and this particular one studied about 0.2 million women over 20-year follow-up period, found that intake of at least 400 international units, and one international unit being 10 micrograms of vitamin D, was associated with 40% lower risk of multiple sclerosis. And there have been other studies from Australia known as the OS immune studies, which also point out to similar findings. But then there are also observations such as prenatal supplementation in mothers during pregnancy can reduce the risk of MS in children. While there have been some in favor of this hypothesis, some have refuted this notion. Another very, very debatable observation that was made and has been of interest is the month of birth, particularly saying that if a baby is born during spring season, and probably its intrauterine period had been exposed to mostly winters where vitamin D is even further lower in the environment, or sun exposure is very low, these babies have more incidences of MS. Now, this is debatable because there have been several statistical anomalies to these findings, because general population also witnesses certain amount of skewedness towards birth rate in springs. So, so this is still a matter of debate, but Health Canada, regardless, recommends that vitamin D be supplemented in even healthy population, and I think that the values are way higher, up to 1,400 international units, when one stands at a higher risk of multiple sclerosis. What are other factors that can, for example, I would be talking about EBV infection. Now, this is another very interesting candidate that comes up during the talks or conversations when you talk about lifestyle factors in general. What about it? Epstein-Barr virus is a ubiquitous herpes virus. Childhood infection can be very asymptomatic and can go unnoticed, but infection in later life is associated with activation of immune system known as clinical infectious mononucleosis. And I would just like to convey the idea that this infectious mononucleosis, which is kind of an indicator of EBV infection, is the one which is most associated with MS and not the childhood infections. Now, this also points out to a very important observation, such as hygiene hypothesis. That is, if people or a person has never been exposed to a virus such as Epstein-Barr virus, which means that acquisition of this virus infection later on in life would present a bigger risk factor for this person of developing multiple sclerosis. And what are the evidences that we think that relate Epstein-Barr virus with multiple sclerosis, or that link with multiple sclerosis? This was a beautiful study that was carried out in military personnel in the States, wherein at the time of recruitment and during their stay as a military personnel, they are asked to provide samples, critical samples, or serum samples, for them to be analyzed for infections such as HIV infections. And of these cases, several of these military personnel, about 300 or so, went on to develop multiple sclerosis. And then these authors, what they did was they characterized these multiple sclerosis patients into EBV negative MS cases. That is, that in the initial sample, they were EBV negative, wherein they looked for antibodies against EBV as well as nuclear antigen for EBV, which was a negative, but also looked at the samples prior to getting onset. So for example, the second sample as is seen here. Now, what they noticed was if this person, a 19-year-old male, had MS onset in 1999, the prior sample was positive, while this person was initially negative to, or seronegative for EBV. Similarly, if you look at this patient, MS onset was in 1997, while the very first sample was negative for EBV, as was the criteria for selection of these patients. The earlier two samples just prior to onset converted to seropositive samples for EBV, suggesting that there could be a correlation between the two. And when they looked at and summarized their findings of all these EBV negative individuals, all of these individuals had converted to EBV positive or had seropositivity for EBV antigens. But it's a very tricky situation, because even the general population seems to have EBV positivity. Even in fact, in their own cohort, they found that 35% of controls had converted to EBV positive. So it's not always, but I think again speaks to the possibility that there are factors that link with genes or genetic predispositions that can cause further bad prognosis for multiple sclerosis. And when we talk of EBV, it is pertinent to talk about something as important as molecular mimicry. It has been postulated that EBV and myelin basic protein, which share similarity in their structures, can be identified by T cell receptors from an MS patient. Now, this TCR or T cell receptor from MS patients can identify both the DRB restricted, DRB1501 restricted MBP, as well as DRB5 restricted EBV, meaning that if a person ever gets infected with EBV, this TCRs are now predisposed or are suited to identify the DRB15 restricted MVP. And that is probably pointing out to molecular mimicry. And in addition to this, there have been other candidates such as chloride receptors from EBV, which are also identified by T cell receptors in these individuals. And EBV can stay latent in B cells. And this is probably one reason, this is just still an hypothesis, that anti-CD20 therapies could be efficient in treating multiple sclerosis. I was talking about hygiene hypothesis. I did bring it up, how hygiene hypothesis, hygiene could be related to immune-mediated disorders. In fact, this stands out. It's a big area in its own realm. If you think about what hygiene hypothesis conveys, it says that low exposure to pathogens in early life poses a greater risk of immune-mediated diseases. And what do we think of infections in general is virus infections. So virus infections such as measles, mumps, and rubella have been ruled out as negative factors for developing MS. In fact, something as parasites or helminthes have been put forth for this hypothesis. It has been postulated by certain findings that helminthes or parasites which remain in the gut can skew the immune system towards anti-inflammatory. And the reason for this is because they want to survive there. They don't want the immune system to attack them. And that is how they go on to live in the niches such as gut. So the idea is, if a person has been exposed to helminth, would it protect a person from developing multiple sclerosis? And there was this study which was again published in 2011, looked at 12 RRMS patients that were infected with roundworm, and other 12 RRMS patients that were uninfected with roundworm. And if you notice the graphs here, this is a graph for an uninfected RRMS patient where exacerbations have been very frequent and higher, as opposed to individuals that have been infected with helminthes. And this was study followed for about 3 to 4 years when these individuals started getting gastrointestinal problems, and then they got treated for anti-parasites. When they got treated for helminth infections, or when they were treated with anti-parasite medications, the exacerbations again increased even in these individuals, which throughout the course of the study did not exacerbate as badly as when they did when they were given medications for treatment of helminth parasites. Similarly, the EDSS scores were also higher as compared to their prior condition, suggesting that there probably presence of parasites confer an anti-inflammatory environment such that immune activation is under check. And there have been several trials around this idea, including a big whipworm such as Trichuris suis ova. While HINT-1 trial lasted for about 3 or 4 years, did not yield any convincing results. The latest trials around the same idea have also given more confidence that yes, there could be some degree of protection conferred by these helminths. And other infectious agents that have been negatively associated with MS happen to be Helicobacter pylori, a gram-negative bacterium, as well as Toxoplasma gondii. And this protozoan is of interest because there have been also studies suggesting that people who own pet, for instance, a cat, and which are natural hosts for Toxoplasma gondii, are better protected from multiple sclerosis, although recent studies have also pointed out that it's probably not the case. So more studies may be required in the line of these evidences. Talking about other infection, this presentation would be incomplete if I do not talk about diet. And diet plays a major role. And this question often comes into picture, like Western diet versus the Eastern diet, and is this something to do with the prognosis of multiple sclerosis? And to begin with, I will just try to talk about a very basic ingredient such as salt, for instance. What we know from literature is that having high salt diet or high sodium diet can increase the rate of EAE. If you look at this clinical severity, is much higher in EAE when these mice were fed with high sodium, high salt diet. And it's also noted that high salt diet does so by increasing Th17 pathogenic cells, therefore conferring more immune activation and more worsening of symptoms in these patients, in these mice. What about patients? Is this what we see in humans as well? So this was a study that was a 2-year follow-up study on 70 RMS patients and looked at the sodium intake and worsening of symptoms over time. Now, the authors of this study looked at sodium excretion via urine and categorized these subjects into 3 different categories. They categorized them into baseline group that consumed less than 2 grams per day to as high as more than 4.8 grams per day of salt intake or sodium intake. What they noted was these patients who consumed greater salt or higher amount of salt had far higher incidence ratios, as can be noted here, as compared to the baseline levels, meaning that these patients who are consuming high salt may have more severe or increase in exacerbation of disease activity. And their own findings also suggest that these individuals who took high salt diet were more than 3-fold increased chance of developing a new lesion on MRI. So it just gives you an idea, although there have been recent trials, such as the benefit trial for salt, refuting this idea that salt can actually indeed be linked with prognosis of multiple sclerosis. But I think it still puts out a very strong evidence towards that if a person is already predisposed to developing MS owing to its genetic composition, then it is discouraged that the individual resorts to continuing to use high salt diet. And if I'm talking about diet, it is important to consider other components such as phenolic compounds. Now, why talk about phenolic compounds? Phenolic compounds are enriched, are present in all fruits and vegetables that we consume, and they also have anti-inflammatory and antioxidative properties. And as you know, oxidation is another biggest concern that we have to deal with if you're trying to curb neurodegeneration part of multiple sclerosis. So I think I just wanted to bring this slide, which is a slide from my own study, where I looked at a cinnamic acid analog known as hydroxycinnamic acid. Now, what I found in this study was cyanohydroxycinnamic acid decreased the production of inflammatory cytokines within macrophages, and then it decreased overall inflammation in EAE when this was administered 4-day post-immunization. As you can notice that this disease severity is far less than compared to the vehicle-treated mice. Now, this cinnamic acid can be found in common spices such as cinnamon, turmeric, and shea butter. But consuming a whole cinnamon comes with other compounds such as also cinnamaldehyde. We don't know what is the overall effect of cinnamon, but this study in a proof of principle does provide an evidence that certain compounds or phenolic compounds can be anti-inflammatory in nature. And if I'm talking about fruits and vegetables in general, there has been a debate around the diet as such, like would eating more green leafy vegetables confer beneficial effects on the course of multiple sclerosis? And then comes into picture the whole idea about fibers. What about fibers? Would they have any effect on the course of EAE or MS multiple sclerosis? So this study published in 2018 by Gurumurthy Krishnamurthy's group shows that these spontaneous EAE mice, which go on to develop features of EAE spontaneously during the course of 6 to 8 months, when fed with cellulose-rich diet, had far fewer incidence rates than the control mice, suggesting that the cellulose or fiber can, despite it not being metabolized completely in the gut, can render effects on something as critical as inflammation. And they eventually went on to find that cellulose-rich diet decreased the Th1 differentiation, although it had no effect on Th17, both in lamina propria of intestine as well as within spleen, and that it also brought about changes in the gut microbiota of these mice that were fed with cellulose-rich diet. So suggesting that yes, diet may play important roles in prognosis of multiple sclerosis. And when you talk of diet, it is also important to consider the very other spectrum of having diet, that is fasting or eating less carbs, for instance. So are there any evidences about fasting and caloric restrictions. It has been noted and has been followed for many other diseases, not just for multiple sclerosis. Fasting has been proposed, or caloric restriction has been proposed as a very strong or negative factor for pathogenesis in general. What does fasting do? Now, glucose is consumed mostly by brain. About 20% of glucose that you eat is consumed by brain. So what happens when one fasts? Fasting, because there's no enough glucose intake, what fasting does is it rather mobilizes fat stores from adipose tissue, subjects them to lipolysis or beta oxidation of fatty acids. And within liver, they are then converted to ketone bodies such as hydroxybutyrate and acetoacetate. And these ketone bodies can actually cross blood-brain barrier and is consumed by brain. Now, by feeding into TCA cycle or Krebs cycle, they can make sure that enough ATP is generated within the brain. So ketone body does that part. But what ketone body also do is that they're generally antioxidant in nature, that they invoke antioxidant pathways such as NRF2 pathways, and also inhibit ROS production and inflammation in general by targeting inflammasome activation or NF-kappa B activation. So what are the evidences that ketone bodies or fast mimicking diet would confer protection to mice such as EA mice? What we see here is that when mice are given fast mimicking diet, that is these diets which are low in calorie and low in protein, as well as ketogenic diet, which are high fat and low carb diet, they go on to develop far significantly less severe diseases than control EA mice, suggesting that ketone bodies generated from fasting mimicking diet and by just providing ketogenic diet can actually confer protection in these EA mice. And if one looks at corpus callosum of cuprizone treated mice after 5 weeks, this is how demyelinated corpus callosum would look like and when you compare it with naive animals, it's highly demyelinated. But when these mice are now fed with fast mimicking diet or not fed, this is how they would look like. So if you take a note on this side, you would see that fast mimicking diet definitely accelerates myelination in corpus callosum, suggesting that fast mimicking diet or fasting diet or ketogenic diet indeed can help in remyelination as well. And I think it is very important to talk about how other factors such as physical activity would shape up the overall course of multiple sclerosis. And there has been great amount of interest in physical activity over and has been proposed as a crucial modifier of MS prognosis. What do we know about physical activity? In general, if you look at different paradigms such as EAE or demyelination models such as cuprizone, it is noted that exercise in either form can decrease peripheral inflammation as well as decrease neuroinflammation and confer neuroprotection by secretion of factors such as BDNF, NGF, and other factors, eventually leading to decreased neurodegeneration and improvement in clinical disability. And when mice or rats that were subjected to swim activity, that is, they were allowed to swim for every 30 minutes every day, it was noted that these mice that were swimming more had significantly less EA severity than compared to the mice that were not swimming or non-swimmers. And the disease severity was very closely linked with when they were treated with interferon beta. And in our own laboratory, it was shown that when mice are given access to running wheels, voluntary running wheels, it enhances remyelination in models of demyelination. In this case, it was a lysolecithin mediated model of demyelination. And if you take a note of this panel, in exercise, it was noted that this MBP, or a measure of myelination, increased significantly in mice that exercised. And the study also goes on to show how exercise indeed enhanced this by increasing oligodendrogenesis. That is differentiating more oligodendrocytes, which are critical for myelination of these exons. So here is just a overall visual conceptualization of exercise in general. If you take a look at an individual who exercises regularly, and looks at brain volume at the top panel and clinical disability in the lower panel, this hypothesis suggests that this individual who has been exercising will have far lower atrophy in brain. At the same time, this person will have more clinical stability and the disability will be slightly more delayed. And MRI activity would be seen far more scattered and fewer. Again, this is just an idea that is put forth suggesting that exercise is indeed important and can work on multiple modalities of the disease pathology. As I've been talking about all these factors, it is important also to bring up a another interesting idea, thought process around gut microbiota. And I will not be going into details because I know Dr. Lemeroy will be covering this topic in next week's presentation, but I still wanted to make a point that different lifestyle factors, including and as well as host genetics, eventually converge on gut microbial composition. Now, all of them influence what your gut microbiota would look like. And if there are unhealthy lifestyles, it would lead to gut dysbiosis or a disbalance in these pro and anti-inflammatory bacterium, which then can lead to disorders such as multiple sclerosis, or at least in EAE, we know that this is a very critical component which plays a very important role in driving disease pathology. Just as an example, to sort of lead into next week's presentation, I think I just want to talk about one study which was conducted on monozygotic twins that were discordant for MS. In this study, they looked at twins, and one twin sibling had MS, the other one did not have MS, and they collected fecal samples, and samples they found that had different composition of microbiota. And when they orally gavaged this spontaneous EAE mice with the fecal samples from these individuals, they noted that these mice went on to develop higher EAE incidence when they were fed with fecal samples from MS twins. Just giving you an idea that the microbiota changes that are present within an MS individual can be recapitulated even in animal models. And also to emphasize that MS individuals do see gut dysbiosis. So what are other imaging factors? The other imaging factors that may be directly linked with physical activity happens to obesity. We know about stress is very strongly linked to general immune-mediated conditions, and something as important as sleep, which is important for melatonin secretion, has been also brought forth as a factor which is closely associated with MS. And in fact, shift work there have been some studies about how working in shifts or at nights confer people prone to more MS. So now, in the interest of time, I will not be going into details. I would rather be asking questions about if you have on this presentation, but I think it is also pertinent for me to emphasize on the idea that lifestyle factors are not just important for multiple sclerosis alone. Lifestyle factors are crucial for other diseases as well. And I think it is very topical if I describe, just as an example, today's scenario on COVID-19. And now, if you think about COVID-19, what comes to my mind is cytokine storm, which has been postulated to be associated with increased mortality with SARS-CoV-2. Now, I may have switched gears here quickly, but I want to emphasize that cytokine storm, which is an excessive production of pro-inflammatory cytokines, such as IL-1 beta, IL-12, and other chemokines, CCL12, if they are produced in higher amounts, it can lead to organ failures, which is what is seen in these patients who undergo or who experience or who have been infected with viruses. Just to bring it as an example, what it was recently reported was vitamin D levels, as can be seen in this. If an individual had higher vitamin D levels in its serum or plasma, this person went on to secrete less C-reactive protein, which is used as a marker of acute inflammation, meaning that people who had higher levels of vitamin D in their serum were prone to less undergoing cytokine storm, and they have had a better survival rate than patients that had lower vitamin D levels in their plasma. I just wanted to bring this out as an example to emphasize that lifestyle factors in general are crucial. And and even if you don't have a propensity for or predisposition to multiple sclerosis, as such, it can confer, these can confer or can induce more pathogenicity if a disease scenario arises. And I would leave you with this question that what if we'll practice in the highest standards of hygiene in the long run, enhance the risk of individuals susceptible to autoimmune conditions. Just as a take home, I would like to just have this slide saying that MS is indeed influenced by gene environment interactions. And this is where I would like to take questions now. Thank you.
Thank you, Deepak. You can stop sharing the slides so that you can see the chat box. And I see a lot of the questions have been answered by other members and experts.
Okay.
So if you can scroll down, and there's one question left from Salma, if you would like to answer that.
I'll just go through them. Has any research been done to test the risk? Yes, in PubMed, there's no risk articles. When I check vaping and multiple sclerosis, I just want to comment that there was a talk at the NMS conference in December that the speaker actually explicitly talked about vaping in general. And there were, I don't remember clearly the data, but she emphasized on the fact that even vaping can be a very strong lung irritant. And therefore, it is also a very strong prognosis factor or risk factor for multiple sclerosis. Jeff, comment on how Latitude and MS seem to overlap with global distribution of other autoimmune allergic diseases. Is there a correlation with MS with hygiene hypothesis versus preliminary? So I think when you talk of Latitude effect, yes, there are effects such as allergies, for instance, generally speaking, arthritis. I'm not explicitly looking into what other diseases are exactly correlated with Latitude effect, but if you talk about Western lifestyle and Latitude effect, allergy comes to mind. Is there any correlation of MS with hygiene hypothesis and prevalence of ailments? Yes, there has been, and this is actually how it started, that countries that had higher parasitic infections like down south or closer to equator did not have as much MS incidences or prevalence, but then the prevalence of helminth infections in those countries is higher. And if you notice the very first report around this came from an Argentinian group that I presented. And there are other groups that tribes, for instance, they have looked at that certain tribal areas or regions do not have infections such as do not have multiple sclerosis in their community as much because there is very high prevalence of helminth infections in those communities. Ben asks, are the MS risk factors actually associated with other immune diseases as well, or strictly MS? Yes, other diseases as well. But when studying MS and you want to look at genetic composition of these individuals, it is pointed out that these have very high correlation with certain HLA haplotypes. But this is important because HLA 1501 is critical for presenting MBP. Now, if these individual HLA haplotypes that are described are associated with other diseases, I'm not entirely sure. But I would also like to talk about how not just these haplotypes are indeed important for MS. And as I also mentioned during my presentation, that certain groups, for example, Spanish groups may have other HLA that can predispose them to MS. So it's not exactly always these particular HLA haplotypes. There could also be other haplotypes that predispose. If having those haplotypes that predispose to MS would also bring other autoimmune diseases, I'm not sure about that. Oh yes, okay, so this has been answered. HLA is also affected by HLA haplotype. Okay, and what do we know the mechanism by which parasites mentioned have an anti-inflammatory effect? If so, it would be successful to fabricate drug to mimic this mechanism. In fact, this is a great point. Do we know mechanisms? So there's this study where they looked at soluble factors from parasites, or secretory factors rather, I should say, and they have found that mere injecting those secretory factors from parasites actually conferred protection in EAE mice. So I think this is where people are heading. This is where they want to know what are the factors, such as, I think there was one factor on the slide, I don't remember it specifically, but yes, this is where the interest now lies in. Parasites secrete factors that promote regulatory T cells and suppress inflammation. Clinical trials using direct have been done to treat. Okay, you have answered this. Perfect. Have antioxidant-rich foods been found to have any effects in MS? Not to the best of my knowledge, if food-rich diet, but I think epidemiological studies or demographics do kind of point in that direction. Like certain communities tend to have more diet rich in, say, fiber or antioxidants, have less incidences of MS. But if directly antioxidant rich foods have been tried in MS, I'm not entirely sure of that. Can you talk a bit more about stress as a lifestyle factor, especially geographically, if stress levels are generally higher in North America compared to places like Northern Europe? Good question. I don't know the stress index, if that is indeed the case. In fact, if I think of other countries, such as Japan, for instance, let's take an example, which is also a very highly sleep deprived country. I think stress levels could also be correlated to that, but doesn't see as much prevalence of multiple sclerosis. So I don't know how to answer this, first of all, and if I can bring stress into picture, clearly associating with MS directly. I think, again, it points out to the fact that stress plus genetic predisposition could be something that is driving the disease pathology. Next is high salt diet is usually due to intake of highly processed foods. Yes, did those studies take into account other than ingredients in processed foods could contribute to what they see? No, they have not, but it's a good point. They have not. Such studies are difficult to control for other ingredients. And moreover, they only looked at urine secretion of sodium. So, but then they also made sure that their eating habits did not change throughout the course of the study. That's all I think they have commented on, but no idea about what other ingredients as a part of this diet was. Considering that fasting and glucose uptake are implicated in multiple sclerosis progression, is there a research done about relationship between diabetes and MS? What relationship between? Okay, yeah, this is a good question. Directly, I'm not sure, but I know that certain like elevated glucose levels in general tend to be more pro-inflammatory. And if one thinks about how glucose can govern inflammation, it is feasible to think that that it could be directly linked to conditions such as diabetes. But diabetes also comes with other conditions such as ketogenesis. That I think is more of a bit of a confounding factor to make out such clear links between the two. Thank you. Did you review the literature on vitamins and MS by any chance? This is a good question. Vitamins as in other vitamins such as vitamin A or vitamin B complex maybe. No, I may not have an answer to this one.
Deepak, really great presentation. Well done.
Thank you.
You know, really thorough, and then linking the epidemiology to mechanism and the immune system, and covering actually many aspects of lifestyle factors. Really, really well done.
Thank you, V. Thanks a lot.
And very clearly presented as well.
Thank you, V. Thanks. I think I may have a few more minutes. Any questions?
Yeah. Well, I think, yeah, no more questions. Well, thank you so much, Deepak, for a great talk. And yeah, everybody else, we'll see you next Thursday with a new presenter. Till then, take care. Bye-bye.
Bye. Thanks for the opportunity. Thank you. Thank you, everyone.